Hello and welcome to this introduction to a cell biology video. I'm Dr. Alid Roberts, and in this video we're going to look at the very beginnings of life on Earth. So how elements became molecules and how these molecules formed the biological macromolecules needed to support cellular life. So when talking about life and its emergence, we need to go right back some 3.8 billion years into the past, when obviously it's not known for certain, but it is thought that something happened resulting in the first steps towards life on Earth. And we need to remember this is only 750 million years after the Earth had formed. Now I guess one of the key questions that needs to be asked is, how did life emerge? What was the catalyst that led to development of life on Earth? And essentially, how did we go from a sterile planet to what we might call primitive life on Earth? Well, it all started with the formation of organic molecules, which in turn led to the formation of organic macromolecules, which, as the name suggests, are large molecules made up of smaller subunits. Now, one of the key catalysts that led to life on Earth was that these macromolecules were self-replicating to a degree. That's to say, at some point, they developed the ability to make copies of themselves, and this makes sense, right? If these molecules didn't have a way to make authentic copies of themselves, then they wouldn't have persisted through evolutionary history, being lost at some point along the way. Now, a second question we need to ask ourselves when it comes to organic molecules is, where did they come from? They haven't always been here, and therefore they must have come from somewhere. And so our best guess, and that's all any of this is really, it's a best guess, is that they were created when molecules in the atmosphere of the early Earth were exposed to sunlight and lightning strikes. And this is thought to have provided the necessary energy requirements to initiate reactions between molecules. Which molecules? Well, it's thought that the primitive atmosphere on Earth contained hydrogen sulfide, nitrogen, hydrogen, carbon dioxide, as well as carbon monoxide, and it is believed in the presence of lightning strikes, these combined over time to form organic molecules, such as amino acids, which are the building blocks of proteins, and proteins have varied functions within biological systems. But how do we know this? Well, it's pretty much an educated guess based on a famous experiment known as the Miller-Urey experiment, where two American chemists, Stanley Miller and Harold Urey, set out in the 1950s to recreate an experimentally controlled environment that mimicked primitive Earth. And so the first thing they did was create a closed system of glass tubes and reaction vessels. This allowed them to introduce known chemicals into the system and record what molecules came out of the system. Now, in order to replicate the conditions of primitive Earth, they needed a few things, the first being water, or more specifically, steam. And so within this closed system, water was introduced and heated so that it would evaporate, mimicking what was happening in Earth's early oceans. Next came the introduction of primitive Earth gases, so things like methane, ammonia, and hydrogen, which joined with the steam and moved through the closed system. And these ended up within the reaction vessel. Now, this area contained an anode and a cathode, allowing Miller and Urey to create sparks between the two, effectively simulating lightning strikes and providing some much needed energy to force along reactions. Now, the gases, after reacting together, would pass through a condenser jacket, effectively cooling the gases to the point whereby they started to precipitate, simulating rain on primitive Earth and completing the cycle. Now, this cycle of heating, reacting, cooling and recycling occurred constantly for weeks on end until they decided to collect some of the precipitate. Now remember, they put specific known quantities of known gases into the system, and their analysis showed that organic compounds, specifically amino acids, were present. Now, these amino acids could only have been produced in one way, from the gases that were already in the system. But how? Well, we can kind of break down the experiment into a series of smaller reactions. Firstly, carbon dioxide could have broken down into carbon monoxide and oxygen. The carbon dioxide then reacts with ammonia to form water and hydrogen cyanide. The oxygen, on the other hand, reacts with methane to reform water as well as create formaldehyde. We can then react the formaldehyde with the hydrogen cyanide that was made earlier, throw in some ammonia for good measure, and create aminoacetonitrile as well as water, which can then react together, forming ammonia and glycine, the simplest and smallest amino acid. And so very quickly, we have gone from some simple chemicals to an amino acid. Now, amino acids by themselves were not enough to promote the emergence of life on primitive Earth. Even when they join together to form proteins, we still need other organic macromolecules to be present, such as fatty acids, nucleic acids, and carbohydrates. Now, together, these four biological macromolecules make life possible based on what we observe around us today, and we have experimentally shown that amino acids can be produced through abiotic synthesis, so the synthesis of new molecules through chemical reactions in the absence of living cells. Now, without going into too much detail, it's hypothesized that fatty acids, nucleic acids, and carbohydrates were also initially produced via abiotic synthesis. And so we're going to look at two of these in a little more detail: fatty acids as a way to make cell membranes, and nucleic acids as a way to catalyze reactions and encode information. And I guess we'll start with the formation of nucleic acids, specifically the formation of ribose nucleic acids, abbreviated to RNA, which is thought to have occurred around the same time as our amino acids. Now, the emergence of RNA, which can be seen here, was another monumental leap on the journey towards cellular life, and this resulted in what we call the RNA world hypothesis, which is essentially a hypothetical point in evolutionary history where RNA nucleotides, so these single molecules we see here, started to come together. And believe it or not, RNA was thought to have emerged before DNA, and so technically RNA was the very first genetic material on Earth. However, today DNA is more important in terms of information storage. Now, as RNA nucleotides became more abundant, they started to form long chains reminiscent of the RNA strands we see today. Now, as these strands of RNA started to form, they developed some unique functions which have persisted through evolutionary history. The first is that RNA could act as a template. By this, I mean it could store information within its structure based on the order of nucleotides along the strand. This ability to carry information meant RNA molecules could form unique three-dimensional shapes, each with its own potential function. But RNA wasn't just a passive messenger. Some RNA molecules, because of their three-dimensional structures, could actually catalyze chemical reactions. This was hugely important on early Earth, where protein-based enzymes didn't yet exist. Anything that could speed up reactions would have been a major advantage. And finally, we get to the most exciting part in an evolutionary sense. If RNA could store information and it could catalyze reactions, what if it could catalyze the reaction of copying itself? That's exactly what scientists think happened on early Earth, and kind of counterintuitively, it can't have been perfect. But those mistakes, which are now called mutations, created variation, and variation is the raw material for natural selection, allowing more efficient replicators to emerge over time. Now, when RNA acts as a template, we typically think of RNA as a strand that is non-folded. But in reality, the order of nucleotides within the strand allow for it to bend and fold into specific shapes, held in place through complementary base pairing. Now, this base pairing can link different strands together or different areas of the same strand, and the result is some uniquely folded shapes, some of which have what we call a conserved motif, which are specialized structures such as hairpin loops, stem junctions, pseudoknots, and even nucleotide bulges. Now, the binding of RNA molecules into these structures will create unique shapes, and therefore they create unique surfaces which may have unique properties, acting as a template that could potentially be recognized by other molecules. And this is essentially when RNA took a large leap towards life, as it resulted in what we might call directed catalytic reactions, whereby one molecule recognizes a template, and once they interact, the reaction creates something new. And so if we wanted to visualize this, we could have one strand, perhaps a non-folded state, and another strand that is folded in such a way that it's able to attach to and associate with other RNA strands. Now, as these two strands start to interact with one another, they will begin base pairing and associating. Now, in this instance, the joining could have induced some form of stress in the template strand, causing it to break. This results in two new strands being formed, which could fold in new ways that they couldn't perhaps before, potentially giving them some unique functions and allowing them to catalyze new reactions that they couldn't before. Now, the RNA molecules or the RNA protein hybrid enzymes we see today, such as ribosomes within our cells, are known as ribozymes, something that is RNA based with enzymatic functions. Now, all of this is great. We have enzymes and ribozymes to catalyze reactions, RNA to store information. However, we don't yet have somewhere to house all of this. We don't actually have a cell. Well, this is where the abiotic synthesis of fatty acids comes in. You see, around the same time, so 4 billion years ago, we started to get the formation of fatty acids, which ultimately led to the formation of phospholipids, which are the key components that define where our cells start and end. Now, it is thought that the fatty acids, which are essentially long-chain hydrocarbons associated with other molecules such as glycerol, which is what we see today. This created a hydrophilic head region and a hydrophobic tail region. Now, the interesting thing about these two regions is that they are opposites. One is water interacting and the other is water repelling. And this can allow the phospholipids to sort of stack together as the two hydrophobic tails interact with one another. Then we have more phospholipids joining the party, and we get what we would call a phospholipid bilayer with an outer leaflet and inner leaflet of phospholipids that have water interacting sections on the outside and water repelling sections within. And so at this point they start to accumulate and form spheres, and as they do, they separate the external environment from the internal environment and whatever just so happened to be encapsulated at the time that this started to form. And this is what we call a protocell. It's another key advancement in the journey to cellular life on Earth. And so at this point, with the abiotic synthesis of amino acids, fatty acids, nucleic acids, as well as carbohydrates as an energy source, they have helped shape the formation of cells and their development on primitive Earth, resulting in life, or so we believe. And after all, this is just an educated guess based on some of the experiments performed by Miller and Urey. And with that, we come to the end of this video. Hopefully you found the content useful, informative, and most importantly, easy to understand. Thanks for watching, and I hope you have a great day.